So let's talk about how vision works. So it's the perception of light in the environment. So it can take the intensity of that light, it can take the color of the light, and interpret it from waves that are coming off from that light bouncing off that object and coming and hitting into our eyes into action potentials. And like for instance here we have an apple. All of the different wavelengths of light are being absorbed by the apple except for one. The one that's being sort of bounced off the apple is the color red. So that's why we see the apple red. If it was a green apple, what would happen is all these colors would be absorbed by the apple except for the color green, and green would be bouncing off. And that's what we would be able to pick up in our eye. So the perception is basically whatever gets reflected off that item is the color we're going to see. And with vision, we can see vision in a spectrum of between 400 and 700 nanomoles, which is going to basically be red, orange, yellow, blue, green, blue, indigo, and violet. So you might have heard that Roy G. Biv. That's the wavelengths that we can see. But if we could see the whole spectrum, we would be able to see radio waves and microwaves and infrared waves, UV light, and then gamma radiation. But we only have this narrow spectrum of 400 to 700 to go ahead and see these waves. So our eye is made up of different parts, and sorry, I wanted to put way more on the slide than I could, so I had to tilt one over. Sorry about that. But basically what happens is your eye picks up these objects, and how it picks up these objects is by having that reflected light bounce back off of that object and into our eye. So if you look at this cup, you can see that the straw looks like it's been sort of bent or at an angle. We know, because we've done this before so many times, we know that the straw is actually straight, but it looks like it's tilted. Well, what happens is when light hits the straw, so let's talk about from here to here first, it's going through air. And there's not much that's going to change that wavelength till it gets to our eye. Down here, what happens is light is still hitting that straw, but it's got to go through all this water. And that water changes the angle of that perceived light, or that light being bounced off that object back to our eye. And because that perception is different, only because the water is in the way, that's why we see that the straw looks bent. If we were looking at it in something else, let's say something that didn't have as much density as water did, maybe we wouldn't see as much of a differential between the one outside the water and inside the water. But because water is kind of dense, especially it's bouncing around, this light wavelength does sort of shift and changes angles. And that's why we see this sort of trick. But what ends up happening is, in our eye, we take this visual cue of this wavelengths coming in. Sorry about that. So the eye contains a series of transparent structures that bend light to focus them to the retina. And we'll kind of concentrate on the retina. That's where the rods and cones are. And they're going to turn that energy, that light energy, into action potentials. Now, different rods and cones, which are going to be those photoreceptors, are going to vibrate at different colors. So some will vibrate because the light that's bouncing off that unit, so if we go back to our color over here, some of the wavelengths will be at a lower color. So something like these dark blues, the purples, they're going to vibrate at this kind of frequency, a little faster. And red will have a different frequency, and yellow will have a different sort of wave frequency. And that's going to tell us how intense that color is. Is it light yellow? Is it dark yellow? Is it going to be blue, or is it that purple color? And that's all going to come from that light intensity hitting our eye. We're also then going to have another photoreceptor tell us about how intense it is, brightness-wise. And those are going to be in the retina. The cornea is going to be the outer layer. It's clear, and basically it's a place that light can kind of focus in. And then the rest of our job is going to be to bend that light into our retina. The aqueous layer, which is going to be that liquid layer, it's going to concentrate that vibrations and, again, focus it back to the retina. The lens will change shape and direction based off of light intensity. If the light is too intense, it'll sort of become small. If the light is too dim, it'll become bigger. And then that visceral humor, if you remember that one, we kind of popped it and then the eye sort of deflated. It gives the eye its shape. It's usually in the back portion of the eye, and it again helps bounce some of that light. If we go to the retina, that's going to be at the back end of the eye. That's where those photoreceptors, specifically called rods and cones, are. And they kind of converge at that optic nerve, which is also where we call the blind spot, and is going to start to send that message up to the brain. So if you remember way back from our skull anatomy, we had this little called the optic canal where the optic nerve came into the brain. And that is going to be where this optic nerve is. That'll kind of curl up through that little hole and go into the brain. And specifically, it's going to go to the parts of the brain where we saw that optic nerve sort of cross over when we were looking at those nerves. It sort of made that X. The retina contains rod cells and cone cells. Basically what happens is the rods are going to sense light intensity. So they will do low light, night vision. They also do peripheral vision. They're going to be most abundant. And then what happens is the cones are going to do color. That's why when you look at your peripheral vision, you can't really tell what color and super shape things are in the sort of edges of your eye. What you can sort of tell is basic kind of color. You could tell basic sort of shape, maybe some blurry image, and that's because mostly at the peripheral vision will be your rod cells. Cones are going to be a lot more closer to the middle of the eye, and they're going to do color vision, acute images, so they'll make images a lot more sharper. What ends up happening is rods will have a lot of these nerves endings, so we'll go ahead and have, this is in the back of the eye, these are the rods and cones. There's way more rods than cones. And what will happen is typically rods will be hooked up to a bunch of rods will be hooked up to one nerve receptor. But each cone is put up to its own receptor. And that's why we get better concentration of colors and things. Where with rods, we're just looking at intensity of light. So we could have 10 of these hooked up to one of these guys, and that's just going to give us basic color. So if we look into the eye, what you'll see is that there's the macula luta, which is basically right around here, and that is going to be where we'll have the most concentrated amount of cones. So we're going to have the clearest image possible. And then right above it is going to be that area where we call the blind spot. The blind spot is there because that's where our optic nerve exists. And because our optic nerve kind of is right here and it sort of would whittle out the back of the eye, there's no photoreceptors there. So what happens is our other eye will be looking at that same image and sort of fills in that little blank that should be there. So it's kind of interesting that our best vision is right here, and then right above it is our blind spot, and that gets filled in with our other eye. The photoreceptors are basically stimulated based off of certain pigments that get excited, and when they move, that movement is going to go ahead and activate that nerve to do those action potentials. So these photoreceptors contain visual pigments, and basically they're going to stimulate during different colors, and those different colors that they get stimulated with will be the difference of what we're looking at. So if it's going to be, don't study any of these numbers, but 420 will be the blue, and then 560 will be the red and the yellows and things like that. And these different pigments that are in these photoreceptors will get stimulated at different wavelengths. So when they get stimulated at different wavelengths, they're going to go ahead and change shape. And the thing that changes shape is going to go ahead and say, hey, it's this color or that color. And the thing that changes shape, that little chemical, is called a rhodopsin. Rhodopsin is just a chemical that anytime it's hit with a certain color or sitting with a certain light energy, and keep in mind, this is just light energy. So you're going to have some rhodopsin pigments that are going to get stimulated at 420. You'll get some rhodopsin pigments that will get stimulated at 500. You'll get some at 530, some at 600. So that's how we could tell these different colors, basically by which rhodopsins are being stimulated. And then what will happen is the light sensory information will come in and send that to a receptor. That's going to go ahead and, don't worry about these G proteins and these kind of things. We'll actually cover those in 202. But what's going to happen is we're going to turn that light energy, move it with that chemical, rhodopsin, and turn that rhodopsin into electrical energy, which is going to activate our action potential to eventually get it up to our brain. So we can go ahead and be able to look at this different colored light spectrum and be able to tell which colors things are just based off of their wavelength and how we can signal that to the brain. And that's going to go to different areas to excite. It's red color, green color, or blue. Great. Our next one we'll do is taste.